finished my 4th month here at UNC Greensboro. And so we all think about the nuclear envelope and nuclear pore complexes in different ways. I've come to think about them in their role in maintaining compartmentalization and the role of the nuclear envelope in maintaining a distinct nucleoplasm and cytoplasmic compartment. And so we typically associate this as being primarily driven by 2 factors. We have the lipids that form the inner, outer, and pore membranes. And then we have the nuclear pore complex, which, while we mostly think about it in terms of transport usually and as a hub, I'm also going to kind of prime you all to be thinking about it in terms of this thought to be a unique proteinaceous barrier to diffusion across the nuclear envelope. So we have this interplay between lipids in the form of the membrane here, and we have the nuclear pore complex here in the form of a protein barrier. And this is all well and good in interphase, but of course, during mitosis, you need cytosolic cytoskeletal machinery access to chromatin to segregate chromatin. So human cells and metazoans broadly take the strategy of simply disassembling the nuclear envelope. I'm oversimplifying here, of course, but really driving home the point that nuclear compartmentalization is lost so that microtubules in the form of the mitotic spindle can access chromatin. And once that is occurred, and once we have kind of pulling apart there of chromatin, we can reassemble the nuclear envelope. And so 2 things have to happen here very quickly. You have to reestablish the nuclear envelope and seal that membrane. And in the metazoan context, you have to reestablish the 2 compartments, the nucleoplasm and the cytoplasm. And so I've gotten really interested in how this occurs, how you build and maintain a compartment, and how you seal the membranes here. And the way I've chosen to investigate this is through a more tractable system, and that's through the fission yeast S. pombe. And on the face of it, it might look a little bit different, and that's because in pombe, the nuclear compartment actually maintains compartmentalization throughout. You can see that blue of the nucleoplasm here being maintained. And so fission yeast have evolutionarily adopted a different strategy for allowing their cytoskeleton access to the nuclear plasm. And so if we zoom in on this little magenta dot at the ends of the spindle, of the mitotic spindle here, you'll see the yeast microtubule organizing center, the spindle body, gets transiently inserted into the nuclear envelope to facilitate construction of the spindle. In late mitosis, the spindle body is extruded into the cytoplasm, and now you have a single hole in the nuclear envelope that needs to be sealed per daughter nucleus. So it's a much more facile system for looking at nuclear envelope sealing compared to the kind of asynchronous dozens of sites that need to be sealed in metazoans. And what's also interesting is that despite this large, hundreds of nanometer size hole in the nuclear envelope, fission yeast still maintain their nuclear compartment. So how is that maintained throughout? And so that's going to be was the driving questions of my work. So how do fission yeast cells maintain that nuclear compartmentalization and then eventually seal their nuclear envelope? And just to drive home kind of how conserved this kind of sealing is, if I show you this kind of electron microscopy that's been collected by other previous work looking into this. If we have on the left here, pombe, fission yeast, and on the right, we have human cells, cultured HeLa cells. You can see how the nuclear envelope shown here in blue is very similar in both cases in the late stages of mitosis. Here, we have the single hole in fission yeast with the spindle hole body having been extruded. We can see microtubules in the nucleoplasm, and we can see similarly one of many holes in this case, in the mammalian context, nucleoplasm, cytoplasm, microtubules running through. The landscape looks very similar, just that there's many holes here. So we only have one to deal with in fission yeast, much more tractable. And so in looking at what could be driving sealing of membranes here, the obvious candidate, based on just their conserved role in nuclear envelope sealing and by work from us and others, probably many who are here, looking at nuclear envelope remodeling, was the escort family of proteins. And this is just a non-exhaustive, albeit laundry list of functions in the cell that the escorts are involved in. Really, this fascinating, yet still, I would argue, mysterious membrane remodeling machinery, primarily driving membrane fission, and in some contexts fusion, that we chose to kind of investigate that perhaps they're what's driving nuclear envelope sealing. And so we did this by doing live cell fluorescent microscopy of tagged escort proteins. And that's what I'm showing you here, starting with the unique escort-2-3 hybrid protein, CHIM7. And so from left to right, I'm showing you stills from an asynchronous population of live cell yeast going through mitosis, which is then resolved on the right side of the slide. And in green, in the merge, and shown here in grayscale, we have CHIM7 tagged with GFP, and then we have spindle full body and the spindle itself labeled here, shown in grayscale, and in the merge magenta. And what you can see is as mitosis progresses and as it's resolved, we get recruitment of this escort protein CHIM7 to the end of the spindle. This was kind of the first evidence that implicated escorts in this process that we were looking at. And I've shown, I've displayed to you for you here to kind of get an idea of when this recruitment of CHIM7 peaks, because there's a whole host of escort proteins that, again, their function's a little bit redundant, a little bit mysterious in how they all play together. So of course we first looked at CHIM7 being it's unique to the nuclear envelope, and we're looking at a nuclear envelope sealing site. But we also looked at the remaining half dozen escort proteins in fission yeast. And I'm just showing you kind of a brief kind of overview of the microscopy that we did. Again, left to right, kind of early to late mitosis, and then the resolution of mitosis for 4 different escort proteins. And if you're an escort aficionado, you can pay attention to the names here. And of course, the homologs, orthologs are shown in our paper, which is now published here. But if you're not an escort aficionado, I just want you to take away that there are multiple waves of distinct escorts occurring, temporally distinct waves to the same location. So we've got different needs for different escorts seemingly at different stages of mitosis. But again, further evidence that these proteins are going to be involved in this sealing process. And now that we'd implicated these proteins, we wanted to take advantage of this system because it's really not known how these proteins, which are thought to form polymers, how they assemble in vivo and kind of at a whole of a defined 100 nanometer or so size. So we took advantage of this to do some quantification of the absolute number of escort proteins at the site of spindle full body extrusion using a ratiometric quantification approach using fluorescence microscopy. And that's the data that I'm showing you over here to try and help constrain a model for escort structure or molecular architecture, if you will, at a sealing site. And this is also shown over here with the absolute numbers. And again, I think one of the things I'll come back to and show in models going forward is just the kind of relative ratios here. Again, if you're an escort aficionado, the full data is available in our paper. So we took advantage of that to try and constrain this architecture here for modeling approaches to get a structural idea of these proteins. And so at this point in our work, a model that was emerging kind of looks like this. We have a landscape where we have this nuclear envelope hole caused by spindle full body insertion. During mitosis, as it resolves, we have extrusion and eventually sealing of this hole. And we have now from our live cell light microscopy data, recruitment of a whole host of escort proteins. Again, I'm showing their relative abundance now here. An initial wave of CHIM7, later in mitosis, a second wave of escorts. Interestingly, after mitosis, we get a third and final wave. And then by the following interphase of G2 of the cell cycle, the escorts are gone. And so from this, we kind of had 2 main questions. The first being, are escorts actually sealing the nuclear? Are they required for sealing at the nuclear envelope? Right, this implicates them, but we don't have direct evidence that they're necessary yet. And finally, what was this role for this kind of unexpected recruitment of 2 waves of escort proteins prior to sealing, right? You can't actually seal the nuclear envelope while there's a mitotic spindle running through it. So why recruit these escorts tens of minutes before you can actually accomplish sealing? Why not just recruit them all at the end of mitosis? So we had that in mind as well, but we wanted to actually look at the membrane ultrastructure. And so that first question I mentioned is, do escorts seal the nuclear envelope? And so to look at membranes, there's no better way than looking at them directly by electron microscopy. And as Vasilisa mentioned, I've developed an expertise in correlative light and electron microscopy, starting my PhD and continuing till now. And so that's what we did to target both spatially and temporally cells throughout the rest of this presentation. I'll show you that type of data. So here we're targeting interface cells after mitosis, where we're spatially targeting the spindle body, temporally after mitosis. And on the left here, we have wild type cells, where you can see the nucleoplasm at the bottom of the image, cytoplasm on the top, and you can see the spindle body here sitting on phos with the nuclear envelope, and you can see it's contiguous and sealed. Interestingly, in the case of a CHIM7 knockout, which I haven't mentioned yet, but we don't get any of those other downstream escort proteins, no other waves when we knock out CHIM7, suggesting they all require CHIM7. And when we don't have CHIM7 and have no escorts at the nuclear envelope, in every cell that we looked at, the nuclear envelope had this small hole about 50 nanometers or less in the nuclear envelope that was abutting the spindle body. And this really suggested to us that nuclear envelope sealing truly does require escort. That is one of the functions that they're there to do following mitosis. And so with that sorted, we wanted then to turn our attention to, why are escorts recruited prior to sealing? If they're just involved in sealing, why not just require them right at the very end of mitosis? Why tens of minutes earlier? And so we had a whole host of electron microscopy data, because we were looking at an asynchronous population of cells. They're not synced to mitosis. We're just using CLEM to target different stages of the cell cycle. And so from that data, we looked at our mitotic cells and we looked at late anaphase cells. And here, in wild type cells, here shown on the left, you can see the spindle body. You can see this small hole that's presumably on its way to being sealed as mitosis resolves. You can see these small remaining microtubules. This is very late in mitosis here. If we look at a cell that's knocked out for CHIM7, again, no escorts with the nuclear envelope, that's at the same point in late mitosis, what you'll see is that rather than a tiny hole that's now smaller than the spindle body, it's much larger than this hole on the left here. And that's quantified over here across the cell population. Specifically, I want you to focus on the difference between early anaphase B, where the spindle body was just extruded, where the hole's about the size of a spindle body. Here, in the late anaphase B, we get what appears to be a constriction in the wild type, smaller than the spindle body, whereas you get a much larger dilation of the nuclear envelope in the CHIM7 knockout scenario. And so this really implied to us that there's some sort of grommet-like function for the escorts prior to sealing, that actually they're maintaining a kind of restricted size of this nuclear envelope hole. And without them, you get this massive dilation in the nuclear envelope. And so we wanted to take a look at, what is the functional consequence of not having this, this kind of tight grommet-like function for the escorts. And so we used a fluorescent reporter for nuclear envelope integrity, MGM4, which is too bulky to diffuse through nuclear pore complexes. And what we found is that in both wild type cells and CHIM7 knockout cells, there's no leakage into the nucleoplasm with this reporter. You can see it's excluded from the nucleus. You can see it also happens to be recruited from the many vacuoles in fission yeast, but on the whole, there's no major difference here. So our question was really, what is plugging this hole? And we looked at membranes, right? We looked at kind of whether or not the membrane was sealed. So we figured, well, maybe we should focus on proteins. Maybe there's a proteinaceous diffusion barrier besides nuclear pore complexes during mitosis. And I'm going to show you the data that suggests that there is. So in order to look at this, we used 1,6-hexanediol, which for our intents and purposes is going to disrupt weak protein barriers. And when we don't treat with the drug and then image our cells for about a half hour, we can see our reporter does not leak into the nucleoplasm. You can see that down here as we look at a merge with the nuclear envelope labeled. If we treat with a low concentration of hexanediol, 2.5%, In these culture conditions, we don't see any leakage. If we treat with 5% hexanediol now things start to get interesting. After about 20 minutes, we see leakage in interface cells, presumably as the FG nups are disrupted. But in that same culture dish that we use the 5% hexanediol, where we have these interface cells leaking after about 20 minutes, mitotic cells in the same dish leak faster after about 10 minutes. And this suggested to us that during mitosis, there's another proteinaceous diffusion barrier besides nuclear pore complexes. And so we wonder, does this barrier cooperate with CHIM7? And that's what I'm going to show you here. We first depleted CHIM7 using an auxin-induced degron. We began imaging, and then we treat with that low concentration of hexanediol, which didn't have an effect on the previous slide. And when I show you two movies here, first with an interface cell, wherein we had low concentration of hexanediol and interface cells, CHIM7 depleted, no leakage in G2 cells. In mitotic cells, however, we do get leakage. And watch this nucleus at the top here. We see in a large percentage of nuclei, when CHIM7 is depleted with this low concentration of hexanediol, we see now we get leakage, whereas we didn't when we had CHIM7, suggesting that this grommet is working together with this proteinaceous plug. And so finally, we said, all right, what could be the identity of this plug? The obvious place to look was the spindle body itself, kind of sitting there en face with the hole in the membrane. And I've just been showing it as this magenta blob, but of course it's composed of dozens of proteins. And I'm going to just show you a quick story on PCP1, pericentrin ortholog. And what we did is we took advantage of temperature sensitive alleles that are lose function at higher temperatures in yeast to look at this. So we used this allele for pericentrin ortholog, PCP1, this allele called PCP115. At low temperatures, the protein is functional. At high temperatures, it loses function. And with the single allele mutant, we don't get any leakage into the nuclear envelope. However, with the double mutant, we do, and that's quantified over here, really suggesting to us that the cooperation between CHIM7 and the proteinaceous diffusion barrier is really CHIM7 and the escorts with the spindle body. And the last piece of data that I want to leave you all with is when we did electron microscopy of the single and double mutant, the extreme leakage of ribosomes that we saw. And so on the left, I'm showing you the single PCP115 mutant, where we can see cytosolic ribosomes happily in the cytosol here. We can see we don't see those in the nucleoplasm. With the double mutant now, CHIM7 knockout, with this temperature sensitive allele, we see this massive loss of compartmentalization as ribosomes leak into the nucleoplasm. And so the model that has emerged from this work is really that we have a cooperation between the escorts maintaining a tight constricted grommet prior to sealing, allowing sealing to occur after this. And during that, in cooperation, we have the spindle body mediating a proteinaceous diffusion barrier. And this work is published, so I encourage you, if you want to see a less abridged version, to check out our work on this. This was made possible, of course, by fantastic mentorship from Megan and Patrick, an amazing undergraduate who's second author on this work, Linda, as well as Yvonne, Will, and Elisa in the Lusking lab. And finally, I just want to say that as of 4 months ago, so now it's been just a little bit, I've started my own lab here at UNC Greensboro. It's a fantastic place to do science. I absolutely love it here, and a great department. And if you know of any undergraduates who want to explore the beautiful weather of the South, some wonderful mountains and the coast, and do some great science, send them my way. And you can follow me on Blue Sky. So thanks for taking the time, and I'll take any questions.